Stem cells, the pillars of body regeneration. Stem cells are often referred to as the bricks of life, being undifferentiated pluripotent cells with a remarkable ability to transform into various specialized cell types. This property gives them an essential role in embryonic development, maintaining the health of tissues and organs, and, last but not least, in healing and regeneration processes. They can be harvested from bone marrow or fat tissue and injected to treat various conditions or to stimulate their own production. Why are stem cells important? Embryonic development. In the early stages of development, stem cells are the ones that give rise to all types of cells in the body, from nerve cells to muscle cells. Tissue repair. Living skin, stem cells are responsible for replacing damaged or dead cells, thus ensuring the regeneration of tissues and organs. Therapeutic potential. Due to their ability to differentiate into various cell types, stem cells are being intensively investigated in medical research with the aim of finding new treatments for a wide range of conditions such as heart disease, diabetes, leukemia, neurodegenerative diseases, autism, or rheumatic problems. Applications of stem cells. Bone marrow transplant. Hematopoietic stem cells from the bone marrow are used to treat conditions such as leukemia and lymphomas. Cell therapies. Stem cells are being investigated to treat heart disease, diabetes, spinal cord injury, and other degenerative conditions. Tissue engineering. Stem cells can be used to create replacement tissues and organs. To help the human body multiply stem cells faster, Pro Natura created the Stem Cell 3X Biotics product. It contains only natural ingredients fermented in SCOBY cultures. Cocoa beans, turmeric roots, buckthorn fruits, AFA algae, polyfloral pollen. Bioactive components in cacao, such as flavonoids, have been shown to stimulate the production of stem cells by activating certain genes that stimulate the multiplication of stem cells, improve the function of mitochondria, the powerhouses of cells, with a positive impact on cellular energy production and stem cell health, and reduce oxidative stress, which can damage cells and DNA. Curcumin, the active substance in turmeric, improves the proliferation rate of stem cells, including spinal cord neural progenitor cells, embryonic neural progenitor cells, and 3T3L1 predipocytes due to its anti-inflammatory, antioxidant, and cancer chemopreventive properties. By documenting C. buckthorn's ability to support stem cell mobilization and increase the number of circulating stem cells, a new mechanism of action has been discovered behind many of the health benefits that have historically been associated with traditional Tibetan, Mongolian, and Chinese medicine. Once mobilized, bone marrow stem cells can participate in the tissue repair process and modulate local inflammation, supporting the use of C. buckthorn in general health maintenance. In a clinical study, it was collectively shown that long-term supplementation with AFA algae, blue-green algae, recognized as the most powerful natural cytostatic, increased the mobilization of hematopoietic stem cells due to the polysaccharides it contains. It can also cause stem cells to migrate to where they are needed for body regeneration. The complex composition of polyfloral pollen fermented in SCOBY cultures, which includes a multitude of polyphenols, polyamines, vitamins, minerals, amino acids, enzymes, and antioxidants, contributes to the stimulation of stem cell proliferation through the following mechanisms. It stimulates the production of growth factors, essential for cell proliferation. Stem protects stem cells from damage caused by free radicals and exhibits immunomodulatory effects, thus positively influencing the microenvironment in which stem cells are located. Plant fermentation influences the proliferation of stem cells as follows: increasing the bioavailability of essential nutrients for stem cell health. Nutrients from plants are easier for the body to absorb. Production of bioactive compounds. A variety of bioactive compounds are formed, such as pre, pro, and postbiotics, enzymes, and polyphenols, with beneficial effects on stem cell health. Modulation of the intestinal microbiota. The fermented product changes the composition of the intestinal microbiota, and a healthy microbiota is essential for the optimal functioning of the immune system, and thus of the stem cells. In a new approach, scientists have found a surprisingly simple way to transform mature cells back into a primitive state. Simply giving mouse blood cells a slightly acidic bath through the phenomenon of hormesis is enough to produce so-called pluripotent cells that can develop into any type of cell in the body and thus repair inherent damage. Thus, the vinegar created by fermenting plants with pH 3.5 from the product composition can be the ideal candidate for this approach. For more information, visit our website.